Hello everyone and welcome to the new session of the biology lecture series, and today we'll be discussing the topic respiration. Alright, so in the previous lectures I have introduced to the topic respiration and we studied about the metabolism, what is metabolisms and what are the components of the metabolisms. Then we studied about the types of the respirations. So we studied about the external respiration, internal respiration as well as the cellular respiration. Then within the cellular respiration, we studied about the aerobic as well as the anaerobic respiration. So anaerobic respiration, now we know, is also known as the fermentation, and it is of two types. First is the alcoholic fermentation and second one is the lactic acid fermentation. Then we also studied about the respiration in the lower organisms. Then we studied about the respiration in the earthworm as well as the grasshopper. And then finally, we studied about the respiration in the higher organisms such as humans. Apart from this, we also studied about the respiration in plants, because plants, they don't have any specialized structures. So they respire using the stomata or with the lenticels. Okay. Then in the previous lecture, we were studying about the steps of the steps involved in the aerobic respiration. Okay. So here we will continue the aerobic respiration. In the last lecture, we were studying about the glycolysis. So glycolysis, as we know, is the common pathway for both the anaerobic as well as the aerobic respiration. Okay. So we were studying about the cellular respiration. So what is cellular respiration? It refers to the oxidation of the food taking place inside the cell. That's why it is known as cellular respiration, because it is happening at the cellular level. So it is happening within the cells, and the oxidation of the food, okay, or you can say oxidation of the respiratory substrate, it is taking place within the cell. Clear? Respiratory substrate, I've told you, can be of three types. One is the carbohydrates, within which glucose is the main respiratory substrate as it is readily available. Apart from these, we have fats and then we have proteins also. All right, clear? Now, as this process is at the cellular level, it is known as the cellular respiration. Now, it takes place in three steps. So this we have already discussed. First step is the glycolysis. Then we have the Krebs cycle. And the last step is the electron transport chain. Okay. So we'll talk about Krebs cycle as well as the electron transport chain. Okay. In the upcoming lectures. In this lecture, we'll talk about the Krebs cycle. So in the previous lecture, we have studied that during glycolysis, what happens? The glucose molecule is broken down, okay, finally into two molecules of pyruvic acid. Okay, so from here on, you have to consider two molecules of pyruvic acid, because a glucose is a 6-carbon compound, and here two molecules of a 3-carbon compound known as pyruvic acid are produced. And we have also seen there are a lot of steps and the enzyme involved during the glycolysis. Okay, so you should remember the all these steps and where the ATP production takes place and where the ATP is used. Okay, and you can also study about the enzymes that are involved in the glycolysis. Clear? Now, here, the pyruvic acid cannot enter into the mitochondria. So that's why pyruvic acid cannot take part in the Krebs cycle directly. So first, the pyruvic acid is converted into acetyl coenzyme A. And this process we have already discussed is known as oxidative decarboxylation. Okay, decarboxylation means removal of carbon, and in this process, carbon dioxide is released. So here there is a linking step known as the oxidative decarboxylation. So between glycolysis and Krebs cycle, there is one more step which is known as oxidative decarboxylation. Okay, oxidative decarboxylation. So what happens in during the oxidative decarboxylation? The pyruvic acid is produced. Okay, it is converted into acetyl coenzyme A. Now, acetyl coenzyme A can take part in the Krebs cycle. So, pyruvic acid, this is a 3-carbon compound. Acetyl coenzyme A, this is a 2-carbon compound. Here, a carbon dioxide molecule is released, and in this process, 2 molecules of NADH is also produced. Okay, clear? So, NADH is produced from the NAD. Alright, so NAD, it get converted into NADH. And 2 molecules are synthesized here. 1 molecule of NADH correspond to 3 ATP. So in total, 6 ATP are produced. Alright, from 2 NADH, how many will be produced? 6 ATP will be generated. Here, one more thing happens. There is an addition of the acetyl coenzyme A. So coenzyme A is also added. And finally, what we get? Acetyl coenzyme A. Now, acetyl coenzyme A can take part in the Krebs cycle. Clear? Now, let's discuss about the Krebs cycle. So, Krebs cycle, what is the site of the Krebs cycle? It takes place within the mitochondria. Krebs cycle, we have already discussed. It is also known as the citric acid cycle as well as the TCA cycle. Okay. Now, Krebs cycle occurs during the aerobic oxidation. So, we have already discussed glycolysis was the common pathway for both aerobic and the anaerobic respiration. So, we have seen if this was glucose, then from glucose, first pyruvic acid is produced, also known as the pyruvate. Alright, now what is the fate of the pyruvic acid? So, this is the glycolysis. So, this process is known as glycolysis, and it doesn't require oxygen, or we can write it do not require oxygen, or you can say it is anaerobic. Okay, it do not require oxygen and it is a common pathway. So first pyruvate is formed. Okay, 2 molecules of pyruvate will be formed. Pyruvate is a 3-carbon compound, glucose is a 6-carbon compound. And then now it can either go for complete oxidation. Okay, so complete oxidation takes place during the aerobic respiration. Complete oxidation, and here it can also go for fermentation, which is known as the incomplete oxidation. Okay, so during complete oxidation, what happens? It will enter into the Krebs cycle. Alright, it will enter into the Krebs cycle, and then later on, next process will be the electron transport chain and oxidative phosphorylation. Now, during incomplete oxidation, the pyruvic acid will undergo alcoholic fermentation. Okay, as well as what? It can also undergo lactic acid fermentation also. Examples we have already seen. So anaerobic respiration is also known as fermentation. So there are 2 types of fermentation, alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation takes place. What is an example? It takes place in the yeast. Okay, and it is also used for the preparation of the alcoholic beverage. Lactic acid fermentation, it takes place in a lactic acid bacteria known as lactobacillus. Alright, and it also All right. It also takes place in the muscle tissues. Okay. Or you can say muscle fibers, or you can say muscle tissues. Okay. So whenever you do a lot of vigorous exercise, then there is a lack of oxygen in the muscles. And what happens? The pyruvate is converted into lactic acid. And then this is the reason for the muscle cramps. And later on, lactic acid is metabolized in the liver, and the body eliminates it out. And then you get rid of the muscle cramps. Now the Krebs cycle, the name, the Krebs cycle is derived from the name of the discoverer, Sir Hans Krebs. So Sir Hans Krebs, he discovered this cycle, and on his name, it is given the Krebs cycle. Okay, so this Krebs cycle is based on the discoverer who discovered this cycle, and his name was Sir Hans Krebs. Now, another name for the Krebs cycle is TCA cycle, tricarboxylic acid cycle, or citric acid cycle. Why it is called first TCA cycle? Because the first product formed here is the citric acid. So what happens? Now acetyl coenzyme may have been produced by the process known as oxidative decarboxylation. Then this acetyl coenzyme may, it binds with oxaloacetic acid. Okay, this is the oxaloacetic acid. Clear? Which is a 4-carbon compound. Here it is a 2-carbon compound. And then what will be formed? A 6-carbon compound known as citric acid. So citric acid is formed, which is a 6-carbon compound. That's why it is known as the citric acid cycle also. And the citric acid, it contains 3-carboxylic acid. Okay. Citric acid, it contains 3-carboxylic acid. Okay, that's why it is known as tricarboxylic acid also, because of the presence of 3-carboxylic acid. C means COH group. Now, the Krebs cycle, it brings about the conversion of the pyruvic acid, fatty acids, fats, amino acids into carbon dioxide and water by the oxidation. Okay, so pyruvic acid, we have seen, is produced by the process known as glycolysis. Apart from pyruvic acid, fatty acids, okay, fats, they are made up of the fatty acids as well as the glycerol, and the amino acids. So amino acid combines to form the protein. So all these molecules, amino acid, fat, fatty acid, as well as the pyruvic acid, these are converted through Krebs cycle finally into carbon dioxide as well as water. So all of these molecules, they provide energy to the cell, because during the Krebs cycle, energy is also generated. Okay. Now, this point you have to remember. This is a very important point. Krebs cycle, it is a common pathway for oxidation of carbohydrate, fats, and proteins. So you can see all these molecules, pyruvic acid, fats, fatty acids, amino acids. Okay, what are amino acids? They combine to form protein. Fatty acid, they combine to form fats. And then we have the carbohydrate. So pyruvic acid is obtained from the glucose, which is a carbohydrate. So we can say that the glycolysis is a common pathway for oxidation of carbohydrates, okay, fats, as well as the proteins. Clear? So this is an image showing you that the Krebs cycle is a common pathway for carbohydrate, fats, as well as the proteins. Clear? So here you can see the fats, they are first converted into fatty acids and glycerol. So you know fats, they are made up of fatty acids and glycerol. And you can see then what happens? Fats and glycerol, they are converted into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is also formed during the glycolysis also. Then dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Then finally pyruvic acid is produced, Pyruvic acid converted into acetyl coenzyme A, and acetyl coenzyme A can take part in the Krebs cycle. And during Krebs cycle, what happens? ATP is produced. Along with ATP, carbon dioxide in water is also produced as a byproduct. Now, if we talk about the carbohydrates, so very simple sugars, they are known as the glucose. Glucose are the monosaccharides and they are readily available. So glucose will follow which pathway? Glycolysis. So what happens during glycolysis? Glucose is first converted into glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-phosphate. Then finally, glyceraldehyde 3-phosphate is formed, as well as dihydroxyacetone phosphate is formed. So these two are interconvertible. Then at the end of the glycolysis, again pyruvic acid is produced. All right. Here, you can see from fatty acids, directly acetyl coenzyme A can also be formed. Okay. So here finally pyruvic acid is formed by the carbohydrates. Similarly, if we look at the proteins, they are broken down first into simpler form, which are the amino acids. So amino acid can then be converted into pyruvic acid here. Okay, and then pyruvic acid is converted into acetyl coenzyme A by the process known as oxidative decarboxylation. Okay, so by the process of oxidative decarboxylation, the pyruvic acid is converted into acetyl coenzyme A, and acetyl coenzyme A can take part in the Krebs cycle. Here, which process is involved or which phenomena? Here, glycolysis is involved. So, okay. Glycolysis, we have already seen what are the steps of the glycolysis. Okay, and finally, the pyruvic acid is produced, or acetyl coenzyme A can also be produced directly from fatty acids and the glycerol. Clear? So this image shows you that the Krebs cycle, it is a common pathway. So we can write here that this image shows you that so ultimately you can see everything is entering into the Krebs cycle. So Krebs cycle is a common pathway for all the respiratory substrate, you can say. What are the respiratory substrate? Carbohydrate, fats and proteins. So Krebs cycle is a common pathway for all the respiratory substrate. Respiratory substrate, I have already told you there are 3 types: carbohydrates, fats and proteins. Alright, and what is the respiratory substrate? A substance or a molecule that is oxidized during the respiration and it liberates energy. Clear? Now, during the Krebs cycle, 24 ATP molecules are produced. Clear? This you have to remember, 24 ATP molecules. So, it starts with the acetyl coenzyme A. So, Krebs cycle starts with the acetyl coenzyme A, which is then converted into several intermediates compound with the release of NADPH. NADH should not be P, with the release of NADH as well as FADH. So I've told you in the previous lecture also, whenever we talk about the mitochondria, we'll talk about NADH. ATP is also produced, hydrogen atoms are produced, okay, and then in the end acetyl coenzyme A is regenerated, and this regeneration is very important for the continuation of the cycle. Okay, this is very important for continuation of the Krebs cycle. Clear? So you can see what are formed. First product NADH, second FADH2 is produced, ATP is also produced, and these are all the byproduct, hydrogen atoms and all. Okay, and in the end acetyl coenzyme A is regenerated. Clear? Now this is a flowchart showing you the Krebs cycle. So this is what? This is the Krebs cycle, which was discovered by Sir Hans Krebs. That's why the name of the cycle is kept on his name, Krebs cycle. So we have seen that the pyruvate, okay, which is the end product of the glycolysis, this is first converted into acetyl coenzyme A, and here 2 NADH molecules are synthesized. Then what happens? Acetyl coenzyme A enters into the citric acid cycle or the TCA cycle or the Krebs cycle. And what is happening here? You have the oxaloacetic acid, which is a 4-carbon compound. Okay, so this 4-carbon compound, it combines with the 2-carbon compound known as acetyl coenzyme A. Then what is produced? A 6-carbon compound is produced known as the citric acid, also known as citrate. Clear? That's why the name of the cycle is also citric acid cycle. Now here you can see carbon dioxide is released and a 5-carbon compound is formed known as the alpha ketoglutarate acid. And here NADH is produced. So there were 2 molecules of the pyruvate produced at the end of the glycolysis. So you can multiply everything by 2 here. Okay, so this Krebs cycle has to occur 2 times because 2 pyruvate molecules were generated at the end of glycolysis. So here carbon dioxide release, there is a decarboxylation. So here decarboxylation is happening. Decarboxylation means removal of the carbon dioxide in the form of removal of the carbon in the form of carbon dioxide. So earlier it was 6-carbon compound, now a 5-carbon compound is produced. Now again it undergoes decarboxylation and again 2 molecules of NADH are produced. Now what happens? Alpha ketoglutarate acid is converted into succinic acid, which is a 4-carbon compound. Okay, so here again there was decarboxylation. And because of this decarboxylation, a 4-carbon compound is produced. So you have to note everything here. At which step decarboxylation is taking place, at which step NADH is being produced. Then what happens? 1 molecule of GTP is also produced. Okay, so here you can see GTP is being produced and you have to remember that GTP means guanosine triphosphate correspond to 1 ATP. So here again 2 molecules of GTP will be produced. Again, why? Because 2 molecules of pyruvate were generated. Now the succinic acid is converted into malic acid. Alright, and here 2 molecules of FADH2 are produced. Clear? And we know 1 molecule of the FADH2, okay, it corresponds to 2 ATP. And if we talk about NADH, 1 molecule of NADH, it forms 3 ATP or it generates 3 ATP. So this you have to remember. So finally malic acid is converted into oxaloacetic acid. Here also 2 molecules of NADH are produced. Clear? So in total how many NADH are produced? 2 here, then 2 here, then 2 here. So in total we can say 6 NADH molecules are produced. So 1 NADH correspond to 3 ATP. So here total 18 ATP will be generated. Now 2 molecules of FADH2 are also produced. These are the products of the Krebs cycle. So 1 FADH2 correspond to 2 ATP. So here 4 ATP will be produced. And apart from this, 1 molecule or 2 molecules of GTP will also be produced. So we can say 1 GTP correspond to 1 ATP. So 2 GTP will form 2 ATP, 2 adenosine triphosphate. So if we add all these, what will we get? We'll get 24 ATP. Okay, 18 plus 4 is 22, 22 plus 2 is 24. So during Krebs cycle, we can say 24 ATP are produced. Clear? This was very simple math. Now let's move on. Now next, after Krebs cycle, we have the electron transport chain. Now let's talk about the electron transport chain. The electron transport chain, it consists of a sequence of electron transporters integrated into the inner membrane of the mitochondria. Clear? So it is where are the electron transporters present? They are present in the inner membrane of the mitochondria. So you can say, what is the site of the electron transport chain? It is the inner mitochondrial membrane, because all the complexes, they are present in the inner mitochondrial membrane. Clear? Now, electron transport chain, it transport electrons from NADH and FADH2 to the molecular oxygen. So the electron transport chain, or what is the source of the electron? And you can also say the source of electrons in the electron transport chain are NADH and the FADH2. Alright, and ultimately these electrons are transferred to the molecular oxygen, and molecular oxygen is converted finally into water. So this happens at the end of the electron transport chain. This is also known as the terminal oxidation. During terminal oxidation, what happens? Okay, so this process is known as terminal oxidation. And what is happening during terminal oxidation? The electrons which are released from the NADH and the FADH2, they combine with the molecular oxygen. Okay, and then protons are also joined with the oxygen, and the resulting molecule is water. Now, during this process, protons are also transferred from the mitochondrial matrix to the intermembrane space. Okay, while oxygen undergoes reduction to produce the water. This we have seen. Molecular oxygen undergoes reduction, means it accepts electrons and protons and it is converted into oxygen, and this is known as the terminal oxidation. Now, while the electrons are being transferred from one electron carrier to another electron carrier, a proton gradient is created. All right. So this proton gradient is very important for the generation of the ATP. Okay, this proton gradient is very important for generation of ATP. So because of this breakdown of the proton gradient, ATP are generated. But we don't have to go in detail. All right. So protons, they are transferred from the mitochondrial matrix. So initially they are present in the mitochondrial matrix, and the electrons, they are transferred from one electron to carrier downstream. Then protons are transferred from the matrix to the intermembrane space. Now, again, how can we define electron transport chain? The electron transport chain, it comprises a sequence of protein complexes. So there are complexes which are made of the protein. So these protein complexes, they are present where? They are present in the, we have already seen, in the inner mitochondrial membrane. They are present in the inner mitochondrial membrane. Clear? And there are in total, you can say 4 complexes, and another complex is also present, which is known as the ATP synthase, and sometimes we call that complex as complex 5. Okay. Now the electron transport chain is comprised of the protein complexes, okay, and various molecules tasked with transferring electrons from the donors to the acceptors through the redox reaction. So what is happening in electron transport chain? There is a sequence. First of all, electron transport chain is a sequence of the protein complexes as well as some various molecules. And what is the role of these molecules and protein complexes? To transfer the electron from electron donor to the electron acceptor through redox reaction. What are the redox reactions? They are also known as the oxidation reduction reaction. Okay, they are also known as the oxidation reduction reaction. Means one molecule is being oxidized and another molecule is being reduced. Okay, now these reactions, these involve simultaneous reduction and the oxidation. That's why they are known as the redox reaction, and they are coupled with the movement of proton also. So the movement of the electron is coupled with the transfer of the protons also, we have already seen protons are transferred from the matrix to the intermembrane space, and because of this, a proton gradient is created across the membrane, and the breakdown of this proton gradient leads to the generation of ATP. And this is explained by the theory chemiosmotic theory. Now, a significant portion of the enzymes involved in the electron transport chain are integrated into the membrane, means the mitochondrial membrane. Now, the electron transport chain, it consists of majorly 4 complexes. So, complex 1 is known as NADH dehydrogenase, complex 2 is known as succinate dehydrogenase, complex 3 is known as cytochrome B and cytochrome C1, and complex 4 is known as cytochrome C oxidase. So you just have to remember the name, okay, and you have to remember that there are 4 complexes. Okay, so this is just an additional information that I am giving you. These are the name of these complexes. Apart from this, one more complex is present, okay, that is now called as complex 5. So this complex 5, this is known as ATP synthase. And what is the function of ATP synthase? It is involved in the ATP generation or ATP formation. Clear? This is also present in the inner mitochondrial membrane. This is known as the complex 5. So proton gradient is created. Now what happens when the protons, they move back from the intermembrane space into the mitochondria, then they pass through this ATP synthase. Okay, whenever they pass through the ATP synthase, that leads to the generation of the ATP. That's a whole different mechanisms. No need to go in detail for that. You have to just remember that complex 5, that is known as ATP synthase, it is involved in the ATP generation. Now what happens in the electron transport system? The hydrogen atom produced during the oxidation of the various intermediates during the Krebs cycle, they are first broken down into the protons and the electrons. Okay, and these protons and electrons, after passing through a series of coenzyme and the cytochrome, combine with oxygen to form the water molecules. Okay, so what was the source of electrons? Source of electrons was NADH as well as the FADH2. So these are the source of electrons in the electron transport chain. All right. Now protons, we have seen there is a simultaneous movement of the protons also as the electrons are transferred. So these protons or electrons, they are passed through series of coenzyme and the cytochrome. So what are these? These are the complexes we were talking about, the protein complexes and the various molecules such as coenzymes. So these helps in the transfer of the electrons as well as the protons, and in the end what happens? The molecular oxygen is converted into water molecules, because oxygen is the acceptor of the electrons and the protons, and this process is known as terminal oxidation. Now during these series of events, one NADPH2 molecule, okay, you know, it releases 3 ATP, and one FADH molecule, it releases 2 ATP, which are the products of the Krebs cycle as well as the glycolysis. So during the electron transport chain, or because of the electron transport chain, so we were talking about that one NADPH correspond to 3 ATP and one FADH2 correspond to 2 ATP. So how these are formed? These are formed through the electron transport chain, and this is also known as oxidative phosphorylation. So generation of the ATP, okay, is because of the process known as oxidative phosphorylation. Okay, where the energy produced by the reduction of the oxygen. So oxygen is being reduced because it is electric and it is being converted into the oxygen. So the energy released by the reduction of the oxygen. So oxygen, okay, is converted into water as it combines with the electrons as well as the protons. So the energy released, okay, by during the conversion or during the reduction of the oxygen to the water is used for the synthesis of or for the generation of ATP. So this is known as oxidative phosphorylation. And by this process, one NADP generates 3 ATP and one FADH2 generates 2 ATP. Now here is a note. What is the net gain? Now net gain of ATP molecules during respiration is 38 ATP. So among them 8 ATP were produced during glycolysis, we have already seen. Then 6 ATP were produced during the oxidative decarboxylation, means during the conversion of pyruvic acid into acetyl coenzyme A, and 24 ATP they are produced during the Krebs cycle. And beside this, carbon dioxide and water are produced. So if you add all these, you will get 38 ATP. So you can say during the aerobic respiration, 38 ATP are produced. Clear? So this you have to remember. Okay. Now you know about the Krebs cycle, you know about the electron transport chain, and you know about the glycolysis also. So you can now, after this lecture, you can now tell about the steps which are involved in the aerobic respiration. Okay. Hope you enjoyed this lecture. We'll talk about rest of the topics, and we'll also talk about some of the questions that are related to this topic in the next session. Thank you very much for listening.